Okay, sup guys? So, the reason why you're here is probably because you need to learn some fucking biology. And since you're here, you most likely don't know what the fuck is going on in class. So I'm here to fix your damn problem. Okay, so I'm just gonna go through the slides that she made for the class itself and help explain everything on each slide, basically. So let's begin. Okay, so probably the first most important thing is, why do we give a shit about cell signaling? Like, who gives a shit? Like, oh look, a cell can talk to another cell. Oh my goodness, that's beautiful. But really, why do we care? Well, it's the reason why everything works. Your brain works based on its cell signaling. Everything in your body is based on cell signaling. The reason why you can just your reason for holding in your urine is caused by different hormones in your body that are forms of cell signaling. Without cell signaling, nothing would function and you would die. And the only things that would be able to survive would possibly be single cell organisms, and even some of those can't survive without interaction with others. So that's why we care about cell signaling. But then, yeah, that's the reason why we care about cell signaling. Okay then, so going on from that, different things like yeast and bacteria, they use it to act similar to multicellular organisms. Like yeast can use them to mate, and bacteria use it to aggregate as spores when there's not enough food, basically. And then if you don't remember what she said about what caused the fight or flight response in this gazelle, it is the cell signal process of releasing adrenaline into the body. That adrenaline is the more common phrase that's known. Today it's called epinephrine, but it doesn't really matter. You can call it either one you want. Okay, and cell signaling is important in not only multicellular organisms, but also the microbial world. So you see these bacteria right here. When they are out of food, they clump together from going from this thing where they're all spread out to clumping together and clumping together even more. Now, why is this good? This helps to keep them together. Helps them survive. Now, you might be wondering, what's the advantage of becoming a spore? Well, you see, when you become a spore, the ones on the center are probably going to survive, while the ones on the outside are going to probably die. But you see, as long as some of them survive, it's okay. Of course, the ones on the outside probably really don't like being used this way, but sucks to be them. So, this is just a self-defense mechanism just to keep themselves alive. Okay then, that's not important. That's not important. Not important. Cool. So, one of the ways to classify cell signaling would be based on its distance. Look at this one, for example. This would be considered local. Why? Because, as you can see, there's a transmission from this original cell over to the next cell, but it's pretty close. Like, that's a pretty short distance. That's a pretty short distance. That's a pretty short distance. But then, look at this one. This one would be considered far because it has to go across a large distance. And this one kind of makes it look shorter than what it can be. What it basically is trying to tell you is it needs to go through a region before it can get to the end. In this case, it has to go through a blood vessel. But let's say this blood vessel is a foot longer. It could go all the way from this endocrine cell over here all the way to the target cell 1 foot away. So it can be pretty far away. That's the reason why it's called far. And this would also be a local one because, as you can see, it's a very close distance. And this one is actually very special because this is nerve cells interacting. This is neurons interacting with each other, which is how your brain works. And if it didn't work, you would die. And as you can see, the neuron from the axon releases neurotransmitters to the Oh, there's no dendrite. Interesting. It sends it over to the next part of the nerve cell. This is a very simplified version of a nerve cell, but we can talk about a nerve cell whenever we get onto that point. So basically, it's a transfer of neurotransmitters from one area to the next, and it's very close. And then the final one that we can talk about is direct contact, and it's pretty self-explanatory. They're directly touching. If you don't understand why it's called direct contact, god damn it, I don't know how to help you. So let's go on to the next one. Okay then, so we want to describe the free again. So what were the free? They were far, local, and crap. You see, I'm not good at biology either. God damn it. What is the other one after local? Yeah, I can't remember. Sorry, guys. Lol. It's direct contact. There we go. Direct contact is the third one. And if we want to give examples of each one, the endocrine system's means of sending things from, like, let's say, the brain in the pituitary gland all the way to the foot. That would be a very far distance, so it's far. If a neuron is communicating with another neuron, that would be local because neurons talking to each other is very short distance. And direct contact would be like two plant cells with, I forgot what those connections are called. We'll probably learn about them later, but there's a way for cells to have, like, the cell itself will, like, have holes in between the two of them, and it allows them to be directly touching with each other. We'll probably talk about that later, but I don't remember what that's called. Sorry, guys. Okay, now, these are the three stages of cell signaling. And if you need a more in-depth about this will probably be also shown in my other videos. And if you need those videos, those haven't been posted to the internet, so just email me and I'll send you the link. I'll, like, share it with you on my Google Drive. I'll share my Google Drive of those videos. Okay, so basically reception, it's where the thing goes into the other, it goes onto the surface protein. The signal molecule goes into the receptor. And it makes sense because you see receptor, receive. Yeah, I know. It's really retarded. Anyways, then there's also the ligand. The ligand is just basically the signal molecule. It goes here, done. And then it's transduction. Okay, just remember, transduction is all the crap in the middle before the response, after the reception, but before the response. It's all that crap. And then we go to the response, which is basically activating whatever cellular thing that needs to happen. Let's say transcribing some DNA or creating some cytoskeleton, which you guys should know is made of actin. Okay? Yeah. Better review that stuff if you want to do well on the AP test, guys. And then we had to look at all those different types of receptors. Remember, they're on those videos. I talked about them in quite a bit of detail on those videos. If you need help, just email me and I'll send you my videos on those 4 types. Okay, so I talked about this in another video, so I'll just going to skip this. I talked about that in the other video, but I'll give you a short review. The signal molecule goes into the receptor and then activates the G protein. GDP goes onto the G protein. The G protein then goes to the adenyl cascade. And then ATP reacts with that and it creates cyclic AMP, and then makes a protein kinase. Cool. Oh, now we get to talk about protein kinases. They're basically cause more chain reactions afterwards. As you can see, they phosphorylate other proteins and enzymes, which usually activates them. So basically what they're saying is it can cause other proteins to activate, but they also can deactivate those proteins. That's basically what it's saying. Okay then, and then have to have a way to turn off the response protein phosphase, remove the phosphate group, and inactivate the protein to shut down the response. Basically what they're saying is they can also shut down proteins in the most simplest terms. Okay. And basically doing both of these is how the activities of the cell are balanced. Cool, so this is a diagram of basically what she was talking about. I mean, I was talking about, wow, I'm just so out of things right now. I'm sorry, this is like 12 a.m. in the morning. I don't even know why I'm making this video for you guys. Okay, so here is the signal molecule. It goes into a receptor. Yeah, we already know what the hell that is. It goes into this activated relay molecule, and then it goes into this kinase. And then it goes into this kinase, and then it goes into this kinase, and then it actually finally activates the damn protein that really needed to happen. But this is called phosphorylation cascade. Notice how it kept going down and down. But this is kind of simplifying the view. If you don't understand why it's called a cascade, it's because it's actually becoming bigger. But this picture itself doesn't really show why. So let me just get a picture of it where it has more of it, and it would be a better explanation. Let's look at this one, okay? Basically, ignore that. That's not that important. Basically, the signal goes in. It causes a protein. The protein then maybe activates 2 proteins, and then that protein activates 4 proteins, and it just keeps going on and on and on, and you can have thousands. You used to have only one, it now has thousands. Obviously, that's going to cause more change in the cell because there's more things activating the cell. Okay. So, what is the point of the phosphorylation cascade? Basically, the point is to amplify. That is the point. It's to make a signal that used to be pretty small, very big. You might be wondering why you need to do that. Well, let's just say, for example, your nerves. Okay? Your neurons are based on a principle called the all or nothing principle. And basically, the all or nothing principle is basically it either gets over the baseline or it won't happen. So let's look at it. So basically, there's this baseline. It has to get over the baseline. If it doesn't get over the baseline, it will not activate. And if it doesn't activate, well, then the nerve just doesn't do anything. Oh, here we go. This is a great picture. You see? Doesn't get over the baseline, so it just doesn't do anything. Doesn't get over the baseline, so it still doesn't do anything. But finally, it gets the baseline, and it goes over. Oh my goodness, right? It's beautiful. It's capable of finishing it. So basically, that's one of the reasons why you might want phosphorylation cascade, so that you can get past this threshold and finally activate what you need to get done. Alright? Alright. And what does phosphorylation usually do to an enzyme? It basically activates a lot more of them, if I'm correct. I'm actually not so sure about that. Sorry guys, I'll probably have to look that up right now. What does phosphorylation do for an enzyme? Okay, so basically phosphorylation basically the phosphorylation can activate or inhibit the enzyme. Oh wow, that's amazing. I totally didn't know that. Thank the Lord I didn't know that. I mean, we were just talking about earlier how it can be used to activate or deactivate. Wow. Thanks, buddy. Really helpful. Anyways, basically what they're just repeating what we said earlier. It can be used to activate or deactivate an enzyme depending on whether this thing inhibits it or activates it, basically. Cool, now we get to talk about second messengers. Second messengers are basically like the earlier things, like these active protein kinases. But the main difference is they're not proteins. That's the important thing. They're not proteins. They just happen to be some sort of molecule that helps carry along a signal as well, but they themselves are not a protein. That's the only real difference. So, I talked about these in a pretty good detail in my other videos. So, yep, we're just going to skip this part. Ooh, and this is basically an application of what we were talking about. Basically, cholera gets to go into our small intestine and produce a toxin which changes our G protein. Why is that bad? Well, you see, now we just get to release tons and tons of water and salt into our body because basically it opens all our channels and our cells are literally just shitting themselves of all their water, and it's like bad because you see cells without water, they don't really function, and when they don't function you kind of die. Okay then, and then ooh, look at these, the free examples. Oh, look at this, it's so beautiful. This is just basically talking about what is it called, glycogen phosphorylation. This is just an application of it. Basically, we have the reception molecule. It goes in, it causes reception, it causes transduction. Look at this. It like cascades and makes tons of molecules. You see, there used to be one molecule that's like 10 to the 6 molecules. That's a lot of molecules. And basically, with all those molecules, it causes glycogen to be broken down into glucose, which can then be used to, make energy, which is pretty important for you if you want to keep living. Yeah. Well, I mean, I don't really want to keep living, but I mean, it's just my own personal feeling. But yeah, it's for you to allow you to live. It's pretty good. Cool. And this is just another application thing. I really don't care. I'm not going to talk about this. Oh, explain how yeast use mating factors and cell signaling to change their shape. Okay, I guess we did need to talk about this. Wow. Okay then, so basically the mating factor enters the G-coupled protein receptor. It goes through this. The GTP binds to the protein. It causes a phosphorylation cascade. That's cool. So basically, they're making more of themselves. And then they go to the plasma membrane. And then when they go to the plasma membrane, formulin is activated. And when formulin is activated, microfilaments are formed. And why are microfilaments interesting? Well, they change the cell shape because they're what is it? They make the they build up the cytoskeleton within your thing, and basically the cytoskeleton is what gives them their shape. Yeah, cool. Okay, and now we get to talk about response and how they are fine-tuned. Okay, so as we've been talking about earlier, there's amplification, which is basically the phosphorylation cascade. Yep. And the specificity of the response, it basically says the response can be used to just activate one response, or it can activate multiple responses, or it can be used to inhibit or activate a response, all that kind of thing. And it can increase the efficiency of the response. This is usually seen by scaffold proteins, which will be talked about later on. And it can be used to terminate the signal. Cool. Okay, so specificity. Shit. Talk about my pronunciations, guys. Cool. Specificity basically tells us the response of a particular cell to a signal depends on its particular collection of receptor proteins, relay proteins, needed to carry out the response. So basically, all that bullshit is basically saying it can, make one response, or it can make two responses, or these two pathways end up activating or inhibiting one response. So basically, this always causes a response, and this secondary one can activate or inhibit that response. And yeah. Cool. So basically, another thing is sometimes you don't want to get tons of molecules like proteins. So basically, I'm sorry for talking like this. Okay, basically, this is basically another kind of amplification, but this is basically amplifying the response itself. Basically, they have these scaffold proteins, and they basically have tons of these protein kinases which amplify the signal for the response. And then, yeah, they can terminate the signal. Basically, if there's not enough ligand, they're not going to be able to send out a decent enough signal, and it's not going to continue. Cool. Alrighty then. We're done for cell signaling, guys. Congratulations. And if you don't understand something that I was talking about, just leave a comment below. So, yeah. Alright, bye, guys.